Grazie. Grazie a tutti. I'm very happy to be here today, and I thank you all for attending this symposium. And of course, I thank once again all my colleagues for deciding to take part in this seminar and to this symposium. So the literature on the multiple uses of EEG in infancy is known to be burgeoning currently. And today we would like to present you with some results from ongoing studies investigating the possible underlying biological mechanism and maybe also windows of vulnerability early in life. So I will not steal any more time from you and from the presentations. And I'm now very excited to introduce you our brilliant speakers. And just to recap, this is the lineup of our speakers for this morning. So we're starting with the first presentation from Chiara Cantiani, who will give us an introduction on the main critical points and the good practices adopted to study typical and atypical development with EEG. Then we'll have the chance to listen to Judith Gervain and her talk on the role of prenatal and postnatal experience in shaping the development of speech and language processing as assessed using neural oscillations. Then we'll have Marco Bugliatti, who will guide us through the use of frequency tagging as a method to investigate neurocognitive development with EEG. And last but not least, Stefania Conte from the US will delight us with a talk on cortical source analysis in developmental samples. So I just would like to remind all the speakers that we will have about 25 minutes for each presentation, plus 5 minutes for questions right after each presentation. And that's it. I would just give the stage to Chiara.
Hi, everyone. I'm super excited to start this session. So actually, my goal today is just to give an overview and an introduction to try to make the point and explain why we think that infant EEG and developmental EEG is so special that we needed a whole session all devoted to this topic. So when we were first involved in the organization of this beautiful Italian garden, a few of us who define themselves as developmental researchers, we thought that we really wanted to focus a little bit on this topic. So developmental EEG, and even more specifically infant EEG, because it's a broad literature, but still there are a lot of challenges in doing this kind of research, and these challenges make this kind of research different than research that we usually do with adult, so different from adult EEG. And so during the first global session, we had a little bit of spoiler on what I'm going to say now. So Professor Francois already presented his studies on language acquisition done with EEG, and he already anticipated a little bit what the advantages and the challenges are of this kind of research. So since our focus as developmental researchers is mainly development, we need to face some challenges. So we know that infants cannot talk, cannot follow instruction, have very short attention span, have a limited behavioral repertoire, and all these things make difficult to assess some skills in babies. Of course, we have several. During time, a lot of behavioral methods have been developed in order to assess skills in babies, but still, I mean, they can be somehow challenging. EEG is an ideal technique to test babies. He is relatively well tolerated across different ages, from newborns to infants, toddlers, and children. And we can use exactly the same paradigm across ages. And this is something very important, especially if you do longitudinal studies. And also, EEG signals are extremely rich and complex. They give us a lot of information. And what I think is also very important, that when behavioral technique tells us something about the end of the process that the baby is doing, EEG and ERP technique tell us something about the process that is going on into the infant brain, and we all know that, with an excellent temporal resolution. That is really important. So, for example, we don't only know if a baby is discriminating between two stimuli, but we know how he or she is discriminating and when in time it's doing the process. There are some challenges even when doing EEG with baby. So first, let's see what kind of questions we can answer by means of EEG ERP with young babies. Of course, we can answer a lot of theoretical questions that have to do with the how typical brain develop. And we are going to listen to a wonderful presentation later on how we can disentangle the role of early prenatal experience by Judith. But by means of EEG, we can also answer to some experimental question about early markers of disorder and longitudinal outcome. And we can even use EEG ERP to test the effect of early training by using the same technique before and after a training. And according to some theoretical framework, so this one is called neuroconstructivism, and we know that the end point of development is unique. And even if the starting point can be similar, then the end of the trajectory is constrained by several factors that make really the end point unique for everyone. And if the end point of this developmental trajectory is atypical, so if we have a neurodevelopmental disorder at the end of the trajectory, the only way to see where in development the problem started is to study the developmental trajectory, so to assess several time points. And this is what we are trying to do at the Medea Baby Lab, where we have long longitudinal studies going on. We are interested in neurodevelopmental disorder, and we study this by means of this longitudinal design, starting at 6 months of age. And testing babies and kids every 6 months in development in order to assess the typical and the atypical developmental trajectory, because we are testing also babies that are okay. Figurati.
No, beh, cioè, ha un non so che di...
Sì.
È che non c'è nessuno.
Sembra di sì.
Allora.
Eccoli. Grazie. Okay, so yeah, this is our longitudinal study, and really in order to assess the neurodevelopmental disorder, we follow these babies from general population and at familiar risk for neurodevelopmental disorders, so language impairment, dyslexia, autism, and we follow them up until later ages in order to track these typical and atypical developmental trajectories. And our main technique, well, not the only one, but the main one is EEG and ERP research. We are also implementing some early training, and also we use EEG in order to track the changes before and after this early training. So now, let's see what the challenges is when you're doing infant EEG research, and hopefully we are gonna see some solutions to these challenges in this session. So the first challenge that you have is which paradigm to select, because usually we know that ERP paradigms are very long. So in order to reduce the noise, we need to have a lot of trials, to average a lot of trial. But this is a problem with infants that typically have a very low attentional span. Of course, if you are interested in auditory processing, that's a little bit easier because you can use long paradigm. In some cases, you can also work with sleeping newborns. Or if you work with infants, you need to have a good entertainer who make the babies feel happy and stay quiet during the experiment. And in our lab, we use some paradigms that are even 20-25 minutes long, but the babies is enjoying during the session, can look to silent movie or play with puppets, looking at bubbles, and that work. But if you're interested in visual processing, that can be a challenge. And one of the solutions, I'm very happy we have Marco here. He will talk to us about the power of steady-state evoked responses. And he will explain everything about this. So I don't want to steal time for that. Then another challenge is EEG data acquisition. There are even some advantages when you test young babies because they have a thinner skull, they have less hair than adults, and this is a plus when you need to put the net on. Still, some of the babies don't like to put the net, they refuse it at all, and what is important that you have a very short time to do that, because if you spend too much time in net application, then the baby is gone and you are not going to do the test anymore. So there are several systems that we can use. There are some EGI system with saline solution and some gel-based EEG system. And every lab decide which kind of system to acquire to do infant studies. In our lab, we decided for a high-density EEG system with saline solution. So I took these advantages and problem of this kind of high-density EEG for babies from Michel Dehaene presentation at the 15 workshop series that occurred this year, because I agree with all of them. So we have some benefits of high-density EEG with babies, because it's not a problem if you lose some of the channel. You have a lot of them. You can have 64, 128 channels, so it's not a problem if you lose some of them. You can just interpolate them. You can do good source localization if you have high density, and Stefania will talk about this later on in the session. And we can average cluster. If you have a lot of cluster, we can have robust clusters to make our analysis on the data. Still, we also may have some problem, because if we have 128 channels and then we just pick a few channels in our analysis that because we visually like them, that can be a problem. And many studies use this approach, and this is a problem. And even if you do cluster of channels, but some channels have a really bad signal, this is also a problem. So you need to be very careful about that. The reason why we decided for high density EEG for EGI system is that the net application with infants is extremely easy and rapid. So here we have a short movie, you see. I mean, net application is really 3 minutes thing, even less. You see, they have a few here. The net is already and it's done. So it's less than 2 minutes, it's 1 minute probably. And the impedance usually is already perfect because, I mean, you see, this is a 6-month-old baby that has no hair at all, and net application is just perfect and very, very easy. But what happened if we have problem with the net application during the experiment, during the recording? Again, I took this slide from Dave Broman because I really like that. I think it really makes the point. So this is our EEG data acquisition and processing work. And by means of this process, we hope to have meaningful information about the infant brain and cognition. So we answer a very important question. But what happens if we have noise at the beginning of this process? Doesn't matter, we can even apply the best processing analysis, the best algorithm to our data, but still if we have garbage at the beginning, so if we can't acquire good data from the beginning, then this garbage, we will take it on for the whole process, and at the end, the answer that we have from our data is just garbage. So this is something we really need to be very careful, and we have to do with babies. It's very, very easy to acquire bad data. So we really need to be careful at the beginning of the process. Otherwise, we risk to bring this garbage till the end of everything. Then the third and last challenging that, I mean, these are not all the challenges that we have. We have a lot, but I decided to focus on them because we are going to see some to these challenges today. But in order to reply to all the challenges, that we will need to 10 gardens, not just one session. Okay, so until a few years ago, there were no automatic algorithm to accurately process infants' EEG data. So most of the lab just took algorithms that were done for adult EEG, try to apply to infants' data, and most of the labs still use visual inspection. What does it mean? That all the EEG was inspected visually by trained researcher, taking decision about which trial was good or bad. Then, in the last 5 years, I would say we had suddenly a lot of new pipeline of processes that were done specifically for infant and even newborn EEG data. And Velu will talk about this pipeline here, the NEAR pipeline. But I will just give you my own experience using the MAID pipeline. That is a competitor of the MAID, but still, I mean, and the reason why I started using this pipeline is that Nathan Fox contacted us during the pandemic. So in 2020, contacted us, say, "Oh, I saw this paper from your group. It's very interesting. I would like to replicate your result. I mean, I would like to use your data, take your data, and try to reprocess them by means of our pipeline, because we have collected similar data. I would like to process even yours using our pipeline." That was great. I was a little bit scared because, you know, when you have some published data and someone wants to reprocess them, you are a little bit scared. So in this paper, we use this kind of artifact detection algorithm. So we used a couple of tools developed for adults, and then we did manual artifact inspection. So we took all our data and we processed them again with this made pipeline. That I'm not going through detail, but it used the ICA in order to correct to reconstruct the signal instead of just removing artifacts. So this is just a few examples of what happened with the made pipeline and with our pipeline. So here, this is EG Lab, and the color mean these are all trials that we rejected. So a lot based on the two different algorithms or by artifact inspection. So we rejected a lot of trials, more than what the made pipeline did. Here you see another example. You see, I mean, the made pipeline correct the signal, but not perfectly, but still. So at the end, the number of interpolated channels with the two pipelines was identical, but the number of trials, the artifact removal, were extremely different. So we kept around 100 trials, but with the made pipeline, almost the double of the trials were kept. But the good thing is that the results of the whole study were exactly identical. So that was baseline EG, so it's not ERP. But still, the signal was identical, even if, I mean, with the MAID, we retained the double of trials. The results were identical, and even the correlation. So that paper was all about correlation between gamma power in resting baseline EEG with language later on in development, and the correlation remained completely the same, and that was great. So I was relieved about that. But still, I mean, it was a nice example of comparison between pipeline, and I think this is something we really need in the field. So I'm done. So we are going to start this session, and I really hope, I'm very excited about that. I really hope we are going to have a nice discussion. So a starting point to really make some step forward in the direction of practices for good data collection and processing practices in infants research. And for those of you who are interested, there is this international organization that is really devoted to these topics. Let's start the session. Enjoy. And I would like just to thank all these people here who work with Valentina, Caterina, and me, and are the people who really every day faces these challenges. So work with the babies, with their parents, with recruitment, and really what I think that they do the hard work, and it's thanks to them that we can answer to our experimental question in developmental research. Okay. Yes.
So what do you make of the comparison in your pipeline and the main pipeline? Says that you say that it's much better to keep more data, but if the outcome is the same, then the argument goes both ways, right? Like it's much better to, but at the same time it doesn't really matter. So you might as well just stick with your.
Yes, but I would say that after this try that we did, then for the future, I mean, now I'm using the MAID pipeline because, I mean, the results are the same, but you don't have to spend hours in inspecting visually your data. So it's really, I mean, I was not very happy because you see here, I mean, I'm really picky and this is noise to me. So keeping this noise in the data is something that make me feel bad. But still, I mean, if then it's the same. I mean, we had much less trials. So what's the point is, do you want less clean trials? Then it's okay to have a bit more noisy trials. If the results are the same, then okay, let's keep little bit of noise, but without spending hours in inspection.
Right, and so you can even have a paradigm
Yes.
And there, if you more noise, then it will make a difference. But at the same time, you could also say, well, but since it corrects for a little bit, at least correct. So, so I think
yeah.
in a certain sense, it's very reassuring that you got the same result. I think that's always very reassuring. But at the same time, I think this was maybe an ideal test case, and what you want is to test this in paradigms where,
Yes.
for whatever reason, either the babies are extremely agitated because they, whatever, the data is noisy, and or you have very few trials, or maybe in very special populations where even a single infant counts because it's a rare genetic disease or whatever. So I think that's
yes.
Yeah.
Yeah, I agree. I mean, this is was an easy case because it's baseline EEG, so we have a lot of trials. But typically, I mean, some paradigm we maybe have 15, 20 trials per condition. So just one trial with noise may impact the results. Yeah, I agree. And that's why I really think that we need this kind of comparison. So now for adults EEG, there's something going on. So there's the mini pipeline project, the mini EEG lab project. I mean, I think we need something even for developmental EEG. Maybe we need even more for developmental EEG because it's more challenging. So maybe in the future we can do something for that. Thanks.
Other questions?
Where is my presentation? Grazie. Okay. So, okay. So it's okay if I stand up. I don't know if I'll try to. Yeah. Okay. So thank you very much to the organizers for having me here. This is really very exciting. Then I cover out. I'll try that. Right. And so I'm going to keep the presentation at the level of theoretical questions and results, not so much the methods, but then I'm very happy to go into that in the discussion, the coffee break or whatever, to try and see what are the kinds of questions we can address and what is it that we can achieve, and hopefully sort of presenting approaches that are complementary to those of the other presenters. So I work on language acquisition, trying to understand how very young babies start learning language. So not small kids who can already talk, but rather those who cannot yet talk. What is it that they learn to be able to talk? And so when we talk about language acquisition, I'm going to start out from spoken language. This is not to say that language can only be spoken. It can be signed, it can be tactile, etc. But for now, we will stick with spoken language acquisition. And essentially what happens is that there is an auditory signal, a physical signal that carries linguistic information, and this is the input for language acquisition. So we, children, and of course adults, as we are doing it now, need to process this modulated carrier signal, right? There's a carrier produced by the vocal cords, the fundamental frequency, which gets modulated in frequency and in amplitude to carry linguistically relevant information. And so, of course, people have been interested in how adults do this, and there have been models that were proposed to account for how adult speakers are able to, or listeners are able to process speech. The challenge is, in particular, the fact that as we speak, we actually use the physical signal itself is linear. It's at a single level, but the linguistic information is at many different levels. So as I pronounce a sound, that's a single sound, but also part of a syllable, part of a word, part of a larger phrase. So how is it possible that if I say the word, I don't know, EEG, then the first E sound is processed by our brains at the same time as a single sound, as part of a syllable, part of a word, part of a larger sentence. And so in a model proposed by Amélie Giraud, David Purple, and other people, oscillations are now very popular. So they propose that there is a system of hierarchically embedded or a cascade of neural oscillations, the frequencies of which somehow correspond to some of these basic linguistic units. So if you look at the linguistic signal here, then you will see that there are fast oscillations in the gamma range that correspond to the frequency of single sounds, individual phonemes, consonants, and vowels. Then there is a somewhat slower oscillatory band, the theta band, so roughly between 4 and 8 hertz, whose frequency corresponds to the syllable. And then there are slower oscillations in the delta band between 1 and 3 hertz, whose frequencies correspond roughly to the frequencies of larger linguistic units, sentences, utterances. And so the oscillations operate all at the same time to process these different linguistic units. Importantly, also, they are coupled, so there is a nesting or embedding relationship such that the phase of a slower oscillation will determine the amplitude of a faster oscillation embedded in it. And this to say they can't be misaligned, right? They can't just run away. I cannot be processing E as a sound sooner or later than processing it as part of a syllable. This needs to go together, right? And so there is this binding relationship between that nesting or sort of embedding. And so in particular, in this system, so it has been shown that human adults have this system in place. Actually, it's there even in monkeys. So this is not specific to language, it's rather how the auditory cortex is built, and then evolutionary language sort of emerged on top of this. So there is this system, there is plenty of evidence by now. Within this system, theta oscillations, the syllable level, is privileged. This is the most robust signal. Also in speech, syllables are supposed to be somehow the foundations of how we process language. And that's also because oscillations in this range are sufficient to explain speech perception under ideal circumstances, meaning a single speaker in silence, as it is now. And that is because, and here we need to take a step back to understand the speech signal. The speech signal can be decomposed, as I said at the beginning, to two parts. There is the fundamental frequency, so the carrier signal, which is modulated in frequency or in amplitude. And so the amplitude modulation is referred to as the amplitude envelope, and the frequency modulation is often referred to as the temporal fine structure. And it is the case that, so there are algorithms called vocoders that allow you to separate the two. Actually, it's the same algorithm as you have in a cochlear implant. So these have actually applications in the real world. But so these can be separated artificially. And it turns out that for typical adult speakers, if they process speech in silence, not if there is noise, not if there are multiple speakers, but at least a single speaker in silence, then just the amplitude envelope alone, so the modulation of the amplitude of the speech signal, is sufficient to understand speech. So you can reduce the speech signal very much, and yet people understand it, keeping the envelope. And there is now lots of evidence showing that our brains entrain to the speech envelope. So what you see is in red, the envelope of a sentence, and in blue, you see an MEG recording of a response to that same sentence. And so you see that the two are nicely aligned, right? So our brain entrains to the speech envelope, and there are two aspects that can be sort of measured, whether the amplitudes are aligned, right? So whether the peaks and dips have kind of the same amplitude, and whether the phases are aligned, right? So that whether the peaks appear roughly at the same place. Obviously with a time lapse, so our brain sort of follows the speech signal, but once you correct for the delay, you get phase and amplitude entrainment. So our question here was, how does this happen? How does this emerge in development, right? So adult speakers have years and years and years of experience in being listeners and speakers, but of course babies don't. And so essentially the question we are asking here in a series of studies is, how does this, now this is schematically the illustration of the embedded neural oscillations, now it's upside down, so the slow one is up, faster down. How does this emerge through development, and whether envelope tracking in this privileged theta range is present. because of course babies don't have that much experience with speech. And the reason why, so the question is always the usual one, is it there from the beginning, or is it something that takes time to emerge? Experience, especially, not time. And the reason why we thought it's meaningful to ask whether it's there in babies is that monkeys can have it, right? So it's not necessarily something that's related to having language. If a non-linguistic species can have it, then potentially babies might be born with it as well. And we had a specific hypothesis, namely that experience shapes this from a very early stage. Hearing is actually operational from, depending on what you measure exactly, from between the 24th, 28th week of gestation. So fetuses can hear. In particular, they hear their mother's voice, their mother's speech. They also feel it in a vibrotactile way, because of course the vibrations also pass, but I'm going to abstract away from that now. But fetuses are embedded in, or they are sort of swimming in a fluid. They are surrounded by maternal tissues. So actually speech sounds very different in the womb than outside of it. And so here you can see the spectrogram. Is there a pointer? Yes, here you can see the spectrogram of the regular speech signal as we are talking now. So broadcast through air, you see the waveform, the spectrogram, and you see the energy. So it's a time frequency representation. Maternal tissue, so the womb acts as a low pass filter. So you can see that under about 400, 500, 600 hertz, energy is preserved. So the lower frequencies, the fundamental frequency of the voice is preserved, but all the higher frequencies are missing. And so the result of this is that what we call the prosody, so the rhythm and the melody of speech are preserved, individual sounds are suppressed. So it's a simulation, so I'll show you what this sounds like. So if you didn't understand anything, that's exactly what it is. So you could hear it's a human voice, right? You could clearly identify it's a human voice. You could identify it's a female voice because you have the fundamental frequency. You could identify the overall melody, whether it was fast and slow. So this is what we call prosody. But individual sounds are not. So fetuses cannot learn words from this, but they can learn about the overall properties, prosodic properties of their native language, in particular rhythm, right? So the temporal information. And so in a larger project, we are investigating a specific hypothesis, namely that the chronology of infants' experience with speech, so first prenatal low pass filter, then postnatal full band, will actually be the cause of the embedding of the oscillations. So prenatally, fetuses encounter this low pass filter signal, which will fine-tune or align the slower oscillations delta and theta with the rhythms of the native language. Languages have different rhythms. And then only after birth does high frequency information come in. And so this is only then gamma comes online. And so because the others are already low, sort of aligned with the native language, gamma gets embedded in the others. So essentially the chronology of experience translates into sort of the causality or sort of the embedding of the oscillations. And so here is what it looks like now, flushed out in time. So at birth, the prediction is that slower oscillations are aligned to the native language, and then later on, the faster oscillation also gets aligned. And so we are looking at babies sort of longitudinally at birth, looking at the impact of prenatal experience around 6 months and at 24 months to see how the oscillations come online, get aligned, and how that then impacts language development. And so the first study I'm going to present was done mostly, as Chiara very sort of nicely described, the hard work was done by Maria, and a lot of the analysis in collaboration with Ramon Guevara and Maria herself. I had a very little part in all of this. And so here is, I'm going to show you data only from the newborns and the 6-month-olds because we are still testing the 24-month-olds, and also the 6-month-old data is not yet finalized, but the newborn data is. And so this is an actual participant from the study at birth and later at 6 months. The babies at birth really are 1 to 3 days old. They are tested at the hospital. At 6 months and 24, they come to the lab. And our approach was the exact opposite of Chiara. So this is an extremely low density setup with 10 channels. And that's because we were testing them at the hospital. They were lying on their in their crib. So anything that covers the back of the head is just not possible to use because they're lying on their heads. And luckily for us, it's quite well known where auditory and language-related effects are in newborns. So it was enough to cover those channels, and we use a gel-based system. That's because it's compatible with near-infrared spectroscopy, which we very often co-record. So we are constrained to have this system, but actually we were very happy with it because having only 10 channels, that wasn't too time-consuming. So we got really good quality data in single channels. And here is the paradigm. So essentially, remember, what we want to know is how they process speech and whether prenatal experience has a role in that. And so these babies were tested in Paris, so they speak, or hear rather, French. So we exposed them to sentences in French, and we chose a rhythmically similar unfamiliar language, Spanish. From behavioral studies going back to Jacques Mélard's work, Thierry Nazy, Franck Ramu, we know that behaviorally, newborns are able to discriminate languages that are rhythmically different, but not languages that are rhythmically similar. So at the behavioral level, we would not expect discrimination between French and Spanish. Even though French is familiar, Spanish is not, behaviorally, they would not be able to discriminate it. Whereas they would be able to discriminate English from these other two languages because English is a very different rhythm than Spanish and French. Of course, English is also unfamiliar to them. We also measured baseline or resting state in silence before and after. And so what we did was, for various reasons we could discuss later, we gave 100 repetitions of the same sentence to a single child, but different sentences across children. So the results are generalizable beyond the single sentence, but within a child, it's always the same sentence, and there are translation equivalents across the languages. The sentences are sentences from Goldilocks and the Three Bears. So this is what it's on slide.
Les ours habitaient tous ensemble dans une maison. Los osos vivían juntos en una casa. Bears lived all together in a beautiful house.
The 6-month-olds get the same paradigm, except everything is shorter because the newborns are asleep, the 6-month-olds are awake, and of course, as Chiara very clearly explained, you can't keep an awake 6-month-old there in the lab for too long. So everything is cut into half. Okay, so what we looked at was envelope tracking and the oscillations. So first, envelope tracking. This is, of course, cherry-picked. It's our best baby, but still. So this is data from a single baby. These are the languages, newborns, 6-month-olds, it's not the same baby. And so in black, you can see the speech envelope of a specific sentence, and in red, you can see the EEG responses. And so again, you see there is a nice sort of match. The correlation coefficients are written here. They're around 0.345, which is actually, it doesn't look like much, but even in adult studies of envelope tracking, you don't find better. Actually, there are intracranial recordings, and even those are not much stronger. So we were pretty happy with this. This is just a single baby, but here are the statistical results. We did sort of complicated Monte Carlo statistics on it, which I'm not going to go into. I'll only show you the results. So essentially, we looked at amplitude tracking and phase tracking separately. And what you see color-coded are the languages. So if you see a color dot of a specific color, then that channel shows, at the group level now, of course, that channel shows amplitude or phase tracking. Okay, and this is at birth. And so essentially what you can see is that newborns track everything. It doesn't matter what the language is. There's no advantage for French, or maybe for French and Spanish, nothing. They just track everything, both in amplitude and in phase. Okay, that's great. In a certain sense, we were happy with that and not so surprised. We got very surprised with the 6-month-old results. That's what you can see. Sorry, this is it's misaligned because of the conversion from a Mac to a PC. But okay, so what you see is that phase, even at 6 months, is perfectly tracked in all 3 languages. But amplitude is no longer tracked in, surprisingly, the native language French and the rhythmically similar language Spanish. Amplitude tracking remains only for the unfamiliar English. language. So 2 things from this. One is it looks like amplitude and phase tracking are 2 different things because they have different developmental trajectories. Phase tracking looks like a basic auditory mechanism. No matter what sound it is, I'm going to phase align with it. Amplitude tracking is quite different. It responds to experience, at least after birth. And I'm gonna, when I show you the oscillations, in particular theta at 6 months, you will see better or understand better why the English is tracked. It has to do with language development, we think. And so it sounds or it looks counterintuitive because it's the unfamiliar language, but the idea is that it's tracked, again, because it's tracked auditorily and not linguistically, but we'll come back to it. Now the oscillations. So these are the time frequency responses in all 5 of the conditions. So the 2 silence periods and the languages, the full spectrum, or the time frequency. And this is time for me to thank Eugenio because this was done with his W tools and a lot of the emails that we exchanged. So thank you, Eugenio, again. So it looks like normal standard EEG, and what we care about, of course, are the specific cross-linguistic differences in the frequency bands. So what we found in the theta and delta band, remember the prediction was these are the 2 bands that are going to be aligned with the native language prenatally, and indeed what we find in theta and delta is an advantage, so greater power in French and Spanish compared to English, both in theta and in delta. So it looks like, indeed, in the 2 slow frequency bands, there's greater power to the familiar language French and the rhythmically similar language Spanish, as opposed to the rhythmically different one. So this aligns perfectly with our predictions. Namely, there is rhythmic discrimination, right? English behaves differently, French and Spanish stick together, and there is no difference between French and Spanish, as is the case behaviorally. Of course, this is not final proof in the sense that we would need to test a group that's exposed to English, because it could be the case that it's the acoustic properties of these languages that trigger this, and no matter what you were exposed to prenatally, everybody would show this. So we are working together with Janet Worker. We started it before COVID, but then, of course, in Canada, the hospitals were closed for much longer than anything I ever saw anywhere. But now Janet is back to her newborn lab at the hospital, so hopefully in a while we will get English-exposed babies, and then we will be able to see whether the pattern switches, right? So whether they will have more activation for English as opposed to Spanish and French. Another thing I would like to show you is that now we collapse across all 3 languages, and you see that there is greater power in these slower bands. Yeah, sorry. So one thing I did forget to tell you is in the gamma band, we found no cross-linguistic differences as predicted. And again, so you can see that again, it's the slower bands that show greater power to speech in general. And what's interesting is it remains even after stimulation. So I'll come back to this in a short while. So during the final rest, even if it's silence, we actually find greater power in the slower bands. And this is nesting. I'm not going to go into this, but this is essentially the phase of the slower oscillations and the amplitude of the faster oscillations. And where there's a stronger color, a warmer color, then there is a nesting relationship. And you see that there is absolutely no difference whatsoever, which is surprising. So it might be, we followed published pipelines, but it might be an issue of sensitivity or sort of just the noisiness of infant data. The 6-month-olds, very quickly, so this is not final results, but what you see here is there is not very strongly, but statistically stronger gamma activation for French now as compared to both English and Spanish. So the difference is not very visible here, but it's statistically significant. So now the familiarity emerges, right? So at 6 months, babies can discriminate, of course, their native languages, even from a rhythmically similar language. And to our greatest surprise, what we found was totally unexpectedly huge theta suppression. In adults, we don't see this. So this is, we absolutely don't know what this is. So this is why we started testing the 24 month-old group, or rather followed them until 24 months, because we think this is related to the fact that at around 6 months, babies start to learn words. And to learn words, you need phonemes. Syllables are no longer sufficient. Phonemes, so individual consonants and vowels, are what distinguish words from one another. So this is a shift from the syllabic level, so theta, towards gamma, the oscillations in the higher frequencies. And so this is what explains the fact that there is no envelope tracking at 6 months. And you don't see this again here because it's not represented this way, but there is less theta suppression for English, precisely because it's not a language that they can process linguistically, but only acoustically. So finally, for the last 2 minutes, we wanted to see how all this supports actual learning. So we went to our physicist colleagues, so this is in collaboration with the Department of Physics, and the paper just got accepted this morning at 7:30. I got the email, so I cannot not tell you about it, so we're very happy. And so essentially the idea was, how do we find in all of these things? So Pierre was very clear about behavior. You can only see a posteriori things, e.g. shows you the thing in real time. Let's look at learning in real time. What does learning look like in this neural signature? And so working with the physicists, we went to what's called a random walk model. So I'm taking you very far, but we'll get back to the. So essentially the idea is that this is a way to represent if there is memory, so learning in a time series. So if you imagine a walk, you start out from 0 and you can take a decision whether to go left or right. Then you could just take a decision randomly at each point without considering what your previous steps were. So you start out and then you go right or left or right or left, independently of what you did before. Then there is no memory in the system. But these systems could be imagined in such a way that the step you take is such that you remember the previous step and you try to avoid that. So if you went left one step, then the next step is going to be right. Okay? So there is memory and the steps are anti-correlated. Or you could do the opposite. There is memory, you remember your previous step, and the probability of taking the next step in the same direction increases. So there's memory and it's correlated. Once you've taken a step in one direction, you continue in that direction. Now, if you rotate this 90 degrees, then you can look at time series and see how far the excursions or the deviations from 0 are. And of course, if you're not correlated, there are going to be some. If you're anti-correlated, very little. And if you're positively correlated, then quite a lot. So you could look at sort of the size of the time windows of the excursions, plot them, and then look at the exponent of the slope of the linear you get. And so if it's totally random, no memory, you get an exponent of 0.5. If there is a positive memory, then it's above 0.5. If it's below, then there is an anti-correlation. And so we sort of conceptualize learning in this system as finding an exponent that is above 0.5. So there's positive memory in the system. And we looked at our EEG data before and after stimulation, so in the initial silence and the final silence, looking at whether the second silence after stimulation shows more memory, understood in the way I explained to you. So these are the languages: French, English, Spanish, the delta, the theta band and the gamma band. This cannot be computed delta because it would need big time windows that we don't have in these short sentences. And so what you see is in silence 1, the system, so the exponent is already above 0.5. This is well known in human EEG. But what happens, what's interesting, is that there is an increase in the theta band, so the slow prenatally experienced bands. This is all newborn data. For all 3 languages, it's statistically significant for French. So there is an increase in the exponent, meaning there's more memory. There is longer range correlations in the signal, or higher autocorrelation in the signal for the native language in the theta, the prenatally experienced band. In the faster gamma, there is nothing, and if anything, it actually increases for English. So this is a first attempt to try and look at sort of catch learning as it happens in these babies. So it looks like their EEG responses become more organized with longer temporal correlations after stimulation with speech, in particular for the native as you would expect. So with this, I would like to conclude and say EEG allows us to look at, as much as it's challenging, it allows us to establish that the neural architecture to process speech and language is already there at birth. Prenatal experience has a role in this. As predicted, the slower oscillations are aligned or fine-tuned to the rhythms of the native language. And the temporal dynamics change, and EEG allows us to capture that. So we can find a neural signature of learning. And with that, I would like to thank you for your attention, thank my collaborators, the funding sources, the babies and their families, and a special thanks to Marco and Eugenio and István Winkler and Gábor Háden for all the sort of like, help, is this normal newborn EEG kind of emails. So thank you very much.
There questions from
Thank you.
Thank you for very interesting. The last bit that you showed, so the Walker model, if I drop the little bracket, it looks very much to me like a contractionist. Isn't it?
I hope it's not. So my theoretical engagements are elsewhere. So construction is in the, say, neuroconst. Yeah. Okay. I don't think of it this way.
Then what is this?
Yeah. No, so I think this is a neural mechanism. This is neither nativist nor constructivist nor anything. This is just biology, if you wish. This is how the hardware is set up. So essentially, you have neural plasticity that is high at a certain point and then decreases. And so in a certain sense, I guess the theoretical debates are obsolete by now. So I do define myself as a nativist because I would think that for language, there is a system that's hardwired to expect certain types of inputs very strongly. So in that sense, I'm nativist, but I think it's no longer, meaningful to really put this so sharply because, of course, we know that things interact. And so I would think that this really defines biological properties of the brain, in particular plasticity and how plasticity changes.
And in this sense, then everything.
Yes.
I found interesting to look at that rate of frequency.
Yeah.
And find the developmental trajectories different. And there is nothing alpha frequency. How come? Because that is, if you want, that is the mental frequency.
Right. So I didn't show you alpha simply. Right. We did look at it, but of course there are 2 or 3 issues with alpha. One is that the newborns are asleep, the 6-month-olds are awake. So that already sort of makes it very hard to compare across ages. The other is, of course, the shift. So if the newborns are slower, who knows what their alpha really is. And then the third one is alpha doesn't enter into the linguistic models. Even beta that I didn't show you has a function. Supposedly that's the top-down influence, so predicting language. And we do find some things in beta. But alpha just really seems not to have a role, at least according to theory. So precisely because of this different states of alertness in newborns and because it has no role, we're just like, get rid of alpha. Be comfortable and not have to deal with the messy thing. So that, yeah. But yeah, I can show you the data. So we did calculate everything for alpha as well. I just don't talk about it because it's supposedly not relevant. And it avoids these uncomfortable questions of, were they really asleep? Did they wake up during the 20 minutes of the study? Yeah.
I have a question on the theta kind of suppression that you show. And two very different factors came into my mind. One is related to the alpha. In fact, because it's difficult to see. I wonder whether in this suppression it's something like alpha suppression, but at a lower frequency because newborns and 6 months old lower alpha frequency. And on the other hand, this reminds me of an old work that we did on speech processing, in which presented streams of trisyllabic words, and in the contrast between when we presented streams of random syllables, we had this theta amplitude going up.
Yeah.
But then when we presented trisyllabic words and subjects that were able to segment these words, the amplitude of the lower, so of the delta oscillations linked to the word presentation went up, and the syllable frequency went down.
Yeah.
And so
Your 2009 paper. Yeah.
I wonder if that's
I think that's exactly what's happening.
Yeah,
Right. So
it's paying attention not only on the syllables but on
exactly. Right. So I think so. Your first question, if we go back to the data, you will see that there is anyway an initial burst, which is very typical for language stimuli. So I don't think this is just state of alertness across the board, because then you wouldn't, right? And so it's in the theta range now. Whether in 6 months the theta is really there, alpha is another matter, and I don't think that's settled. But still, it's not everywhere. We really get this initial, like, the N kind of thing. So I don't think it's just state of alertness. Now, answering your other question, it's exactly this. I think it's exactly allocation of resources. And I think, indeed, as you say, the syllable at this age, for what they're trying to do, namely learning words, is not relevant. So it's suppressed. And in your case, it's the larger units. I think it's here both the larger units, but especially the faster units. So I totally agree. I think that's exactly what is happening.
Down which neural populations or areas are generating.
No. So, okay. From this data, absolutely not. Of course, with 10 electrodes, there's no way we could do anything about that, right? So all I can do is rely on the literature in animals, in adults, a little bit on babies, and would really strongly hope that this is primary auditory cortex. This is the best. Yes. I would think this is so. So for example, I think the envelope tracking data, just the fact that there is tracking across the board, at least face tracking for all languages, etc., strongly suggests that this is not a language-specific phenomenon; it's an auditory phenomenon. We are running the Maria is running the study with non-linguistic material. So comparing language, monkey vocalizations, music potentially, so to try and understand really how general it is across the auditory domain. So I would think that this is really basic auditory, a lot of it. And only later, so indeed, these are the kinds of things like the theta suppression that are linguistic really, so that they may be elsewhere, but a lot of it is basic.
Thanks for the talk. I have seen that you have presented also baseline and at the end of the experiment. Would you like to discuss a little bit, because there were some differences?
Exactly.
Baseline each.
So, right. So essentially, the first thing is to show that compared to initial rest, when there is stimulation, there is indeed an increase around that frequency, right? So this is even how the adult studies were established back in whatever 2005, 2007, and Genova has done a lot of this. So that's great. And then the point we cared about was looking at resting state after stimulation. And so these are several minutes, right? So it's not just a couple of seconds after. These are 3 or 6 minutes after. So, and indeed, right? So some of the power remains, which I think is somehow a signature of learning or processing, right? So once you stimulate the child with language, something has to happen in the brain, right? Because we think about language development always in these like really large scales from birth to 6 months or whatever, several months. But in reality, of course, they learn from what they hear in the moment, right? And so I think that's exactly what it is, or this is what you're trying to capture. So, okay, I heard my native language and whatever it is that they're extracting from it, that's happening there after you talk to them, not a month later, but right there. And so I think this was the idea, to capture some of these effects. And of course, we don't have a neural mechanism. I would not know what exactly is happening at the neural level, but of course, you know, it's InfantEEG, so you don't know exactly, but at the same time, it's there, right? So you do have a signature of it, and then now we need to sort of try and understand or figure out what really is happening in the auditory cortex, but hopefully something, because it should, but people don't really talk about it this way, right? So we never think of learning really as like, okay, they've now memorized something, right? But that's what we're trying to get. And of course, in the NIRS literature, there are apparent, in particular in Japanese groups, have been doing this with NIRS as well, looking at resting state before and after stimulation. And they also find similar effects in NIRS. So you can really capture that after stimulation with language, something extra is going on, like this processing continued. So thank you so much.
Thank you. The stage to Marco with this presentation.
Okay, thank you. Maybe I'll stay like this. Okay. First of all, I'm very happy to be here. Thank you for the organizer for inviting me and for organizing this wonderful morning on developmental EEG. I hope, as others said, that we can increase this, because I think this is a very exciting sector in human neuroscience. And thank you also to Chiara and Judith, because you introduced a lot of things that I wanted to talk about. So already know that, so I will go faster on some things, and you already have all of this in mind. I also must say that we are pretty in line in the approach that we have. How do we use the methods, and what are the most important challenges to face with developmental EEG? I would say, contrary to Judith, I will really focus on the methods, rather than on the content of the studies that I will present. But of course, feel free afterwards to discuss about the content, which are, of course, the most important thing. So today I will talk about frequency tagging as a powerful method to investigate neurocognitive development with EEG. And I start with like this. Okay, I start with what Chiara was mentioning, in fact. So the fact that we are used, many of us are used to adult EEG data, in which we have typically very young and sort of bionic students that can be attentive for 1 hour without blinking and without moving, and with all their attentions. So all the paradigms that are built on these kind of subjects, we can sort of forget them. And so it is much more difficult to investigate neurocognitive development in infants down to newborns that have, I point here 3 big issues, but of course there are others. And these issues also, some of them Chiara already mentioned. So first of all, these are unconstrained subjects. You cannot tell them not to move, not to frown, not to move their mouth, etc., not to talk. As also the presentation by Belu will show, the problem with movement is that it causes artifacts that are non-stereotypical, so they cannot be corrected, at least not in a clean way. So they're really much worse than any kind of artifact that we have with adults, for which we have blinks, eye movement, that can be removed with ICA, for example. And then the second is also as Chiara was saying, that the subjects are not attentive. I mean, they can be attentive, but for a short time. And this is particularly difficult for visual stimulation. It's easier for auditory stimulation, but still it's a challenge, it's a problem. And these two big issues reduce the time, the duration of your experiment of at least one order of magnitude with respect to adults. So you're really constrained to having, I don't know, 2 conditions, not more, and in a very short time. And also I add, as I work with newborns and very young babies, that there is a third constraint, that is the physiology of newborns and very young infants make it that the neural responses are much slower in some cases than the ones of adults. So you cannot present the kind of fast paradigms that you can present with adults. This is another constraint. So the overwhelming majority of the experimental design used with developmental EEG are event-related designs. So you present isolated stimuli in time, and then you average and you use your measures, be it event-related potentials or spectral perturbations, phase analysis, etc. And here you are assuming that you have your signal of interest and some noise. But this noise, which is also a lot of just ongoing EEG fluctuations unrelated to the stimulus, goes down with the number of trials very slowly, with the square root of the number of trials. So you need many, many trials to have a clean signal. And also another problem is that since you have really a short time, ideally you would like in one condition to be able to dissociate the different levels of sensory, perceptual, and higher order responses. And this is not something that you can do with these designs, because you have the stimulus, and then you don't know which part of the signal that you see is depending on the sensory response or on higher order response. So frequency tagging is a powerful method to address some of these issues. Of course, by sacrificing time to frequency. To explain, I present a very simple example, which is if I present you with a reversing checkerboard, so black squares going to white and vice versa every 250 milliseconds. This is a stimulus that has a very precise frequency of presentation, which is 4 Hertz. And it's a very strong stimulation for your visual cortex. But if you look at the single trials at the onset of the switch between black and white, you cannot see by eye from the single trial the response to this inversion. Why? Because there is a lot of ongoing EEG activity that is unrelated to the stimulus and that covers these fluctuations. So if you reason in the event-related fashion, you average across many presentations, and you easily obtain a P100, so a potential with a clear peak and a clear posterior topography, which of course shows the activation of the visual cortex. But since your stimulus now has a very strict characterization in time, it's a frequency at 4 Hertz, can imagine that the neural populations that respond to this reversal also oscillate at the same frequency. So in this sense, you instead of looking at the signal in time, you look at the signal in frequency, and you typically have a very sharp peak, in particular if you have a relatively high frequency. So as you see here, now you can take a measure which is amplitude of the power spectrum at the frequency of stimulation divided by the average of the amplitude of the power spectrum in neighboring frequencies, and then you obtain again a very similar topography because it's the same source, but with the signal to noise ratio that is in this particular case 5 times the ones that you obtain with ERP. So with this, I hope to convince you that so this is the sort of basic property of frequency tagging or steady state responses, that you obtain a much higher signal to noise ratio because you are much more able to dissociate the stimulus related response from the background EEG that is not related to the response. Okay, my purpose in this talk is really to give you some indications about how I think what are the most optimal ways to use frequency tagging in development. So just a very brief recall of the main tool that we use, which is Fourier transformation. It's probably trivial for most of you, but just to pinpoint what we are talking about. So Fourier transformation is this magic transformation that you put a time series, whatever time series, periodic, non-periodic, no assumption on time series, and then you decompose it into components at different frequencies, and each component is completely characterized by a phase and an amplitude. So, and this is very useful when you have frequency, as this is the case in your system. So I will skip this, and before going into the studies, the few studies that I'm going to present, in fact, the overwhelming majority of the frequency tagging studies employ three measures to measure the response. The first one is, I already showed to you, is the power spectrum. So the power spectrum is the, so you take your continuous time series and you cut some windows, and for each window you compute the power spectrum, and then you slide the window and you compute this power spectrum and you average across all these windows. So the power spectrum only computes the amplitude. It doesn't care about the phase. So it is, I mean, if in two different blocks of the same stimulation, the phase is different, but the amplitude is the same, the power spectrum is perfect. On the other hand, you can expect, or you can assume that the response to a periodic stimulation depends on the phase. I mean, it's phase locked to the stimulation. And in this case, you can first average across these windows, non-overlapping windows, and then compute the power spectrum. In this way, what you measure is the amplitude of the oscillations that are time locked to the oscillation, to the stimulation. And the third measure, you can directly measure the phase. So, okay, I'm going on with this. You can directly measure the phase. So the phase is independent of the amplitude. So whatever the amplitude is, you measure the phase at each time window, depending on the stimulation frequency. And then you're going to see if phases are aligned or not. So in this first case, they are not aligned. I don't know who is, I mean, I can go into details if you want about this representation. And in this case, they are aligned, and intertrial phase coherence is a measure that goes to 0 if they are not aligned and goes towards 1 if phases are aligned. Okay, so I will talk mostly about these three measures. And before going on, just a very brief historical recall on steady state evoked potentials, as they were been called for years. I explain you why, in fact. So, can I use the pointer? Okay. So this is an old study showing, again, I think, reversing checkerboards to adult subjects and with different frequencies. So look at the first. So and this is the evoked potential averaged over the reversals for 1 Hz stimulation. So it's a very slow stimulation for adults. And what you see is that at each reversal, you see the ERP and then nothing. So there is a response, a transient response, and then it fades away. But if you increase the frequency to 3 Hz, 6 Hz, and down to 8.5 Hz, you see that since the next stimulus comes earlier than the decay of the response, there is again a wave and eventually a sort of sinusoid, steady state sinusoid that comes out, and which gives a very strong response. And this steady state gives the name to the steady state response, which in fact was used for investigating the integrity of the visual or auditory system, sensory systems in general, because it was very effective. It is very effective. And in fact, there is a sort of frequency range in which visual stimulations like this the brain gives the strongest response, more or less between 8 and 20 hertz. And for auditory responses, you have higher frequencies around 40 hertz. So if you want to study the sensory responses, this paradigm is very, very efficient. It gives you a very strong response. But of course, it's sensory responses. So then in more recent years, people said, okay, maybe we can use this kind of stimulation to study higher order responses. And so they started using lower frequencies. And so from here, we come back to here, in fact, and this is sort of, let's say, these delta and theta frequency ranges are the ones that I will talk to you about for many reasons that I will go on. Not only because we want to study higher order responses, but also because frequency tagging allows, the ability of dissociating different levels of responses also depends on the ability of using low frequencies. To show you that, so I will present you only 2 studies, and this is again for illustrating the method. 2 studies that I think give nice examples on how to use frequency tagging with infants. So this first study by De Herring and Rossian investigated face categorization in 4 to 6 months old infants. And here the aim is to be able to find a very clear neural correlate of face categorization within the short time of attention of these infants. And in fact, the trick here that we can do with frequency tagging is that they presented a stream of strictly periodic images of anything, any different objects, but every 5th image was a face. Here's the face, here's the face, and here's the face. So the nice thing here is that you dissociate the generic visual response of the system, which is associated with the frequency of single image presentation, 6 hertz, with the frequency of faces, which is 1.2 hertz. And the idea is that if there is a response to face categorization, this will emerge at 1.2 hertz. No other kind of response can emerge at this frequency. So it's very specific. And in fact, that's what they found. If you analyze the peak at 6 hertz, and by the way, here they used the evoked power spectrum. So they average and then they compute the power spectrum. And so at 6 hertz, you see that there is a nice medial occipital response, typical of the early visual system. But then if you look at the response at 1.2 hertz, you have a peak, which is very, very much light lateralized. And then I want to know what Stefania Conte will think about this very lateralized blob. Anyway, this is to show you that frequency tagging in a single block and in 3 minutes of experiment, because this is the time, the average time that infants were attentive to the stimulation, is able to dissociate a visual response from a very specific response. Okay. And sorry, I wanted to say that this is one reason why we need to study what is the best method to analyze low frequency responses, which is 1.2 hertz. So in this case, it's not because the process is slow. It's because the construction, the way that the experiment is designed necessitates to use a very low frequency. But I want to say that also, for example, if you think about what Judith presented, in your case, the issue is the fact that the intrinsic frequency of the process is low. So all the things that I will say about this method also are valid for the auditory processing and speech processing in particular. So now I present the second study. This is a study that we did now a few years ago. And it is, and here you will see that the low frequency really depends on the fact that we are studying newborns. So we expect that the neural responses on newborns are very slow. So we need to present them with very slow stimulation. So here the idea is to investigate whether there is a cortical route to phase, or better, phase-like processing in newborns in the first 3 days of life. And this study takes inspiration from a number of studies that show that, behavioral studies that show that infants have preference for phase-like stimuli like this. So these are the most simple representation of faces that you can imagine, because it's 2 blobs representing the eyes and one the mouth, and the shape of the face, and the contour stimulus is visually equivalent apart from the inversion of this configuration, but this is clearly not a face. So our aim was to say also something about really the cortical sources of this effect, knowing that we know perfectly well how this network is organized in adults and even in older infants, and we wanted to understand whether we could say something about newborns. And okay, so we presented these stimuli in blocks. Here, it's not really frequency tagging. Classical frequency tagging is something like steady state response in the sense that I have one single frequency, and I use frequency tagging just because I want a very high, the maximum signal to noise ratio in these very difficult situations. So I have a tag frequency of 0.8 hertz, very slow, because we wanted to be sure that newborns could attend the stimulus, and that was the case. So, in fact, I can go straight to the response, otherwise I don't have time. So, in fact, what we could see is that the response, so this is the power spectrum. We used the raw power spectrum because we had really not a lot of trials. And what we could see is that in a quite distributed region, we have a higher peak, significantly higher peak for the faces, face-like stimuli with respect to the inverted faces. And what we did also here, and this is also thanks, I'm looking at Chiara, because you mentioning the high density EEG. In this case, we used the high density EEG, 125 electrodes. And since newborns have a very thin skull, we did source reconstruction because it has a better spatial resolution than adults. And we found some overlap with the typical regions that are found in adults for phase perception. The important thing methodologically here is that the average data for each condition is 35 seconds, after, of course, cleaning artifacts, being attentive to fixation times, etc. So this is very short, and we are really at an extreme. Okay, and this is why in the very last part of the talk, I'm going to mention to you 2 very recent investigations, methodological investigations I did for addressing really this problem, which is frequency tagging in the limit of short segments because they are infants, and low stimulation frequencies because of the reasons I just told you. And so this is preliminary data, but I think it's, I mean, there is already a message. So, and also it's preliminary data that have been tested on an adult EEG data set that we recorded while we were recording the newborns with similar frequencies, because we wanted to have really some toy data where we could investigate methodologically what is the best method, and also some simulations that I will probably skip because I don't have time, but you're free to ask me afterwards. And I just want to say that this sort of approach to study, for example, adult data or simulation data with the same kind of tools that we use for infants, I think it's valuable because it gives you some references when you know the golden standard, because that's the problem with this kind of data. So, boy adult EG dataset, 12 subjects, a checkerboard visual stimulation on-off, which is because that's the way we present any stimulus, face, dots, or whatever. And two stimulation frequencies, 0.8 Hertz, which is the difficult one, and 4 Hertz, which is easier because the background EEG is much shallower. And also resting state trials and 25 seconds trials. Of course, it's adults, so they are attentive. So these are clean 25 seconds. And the idea here is to take the 25 seconds trials, consider them as a sort of golden standard. So say, okay, this is the signal that I have, and then decrease the length of the signal that we have, length of the data, to simulate what happens in the worst case of infants, and see which one of the three measures, power spectrum, evoked power spectrum, and intertrial coherence, resist better to this decreasing in data. And so what happens when you decrease? So first of all, at the 25 seconds, you see the power spectrum has a clear peak at 0.8 Hertz. And then for the 4 Hertz data, it has a clear peak. And while at rest, I see nothing. So that's fine. Same thing for the evoked power spectrum. And also same thing for the intertrial coherence. Here I have a peak, here I have a peak, and here I have just noise. So what happens when I decrease? When I decrease, it happens that, so I go on, this is 15 seconds. So at 15 seconds, you already see that, so the power spectrum sort of resists here, and these two resist, and the intertrial coherence becomes increasingly noisy. And in fact, when I get to 10 seconds, which is really short, because 10 seconds, in fact, is 8 cycles of 0.8 Hertz. And so in this case, I have, in fact, 2 phases only. So the intertrial coherence cannot be used anymore, while the other two sort of resist. There is a peak here and a peak here as well. And in fact, I forgot to say that this is with the 1/5 of the cycles of, this is from 5 to 2 seconds. And in fact, at 4 Hertz, something similar happens. So this is not only specific for low frequencies, even if it's worse. And the result here is that, let's take it as a t-value, let's say. So this is the t-value of the 0.8 Hertz stimulation compared to the rest of stimulation for the 12 subjects. And again, here you see at 25 seconds, all the 3 measures give the maximum. I have yes. And when you decrease, in fact, what happens here is that the intertrial coherence goes down for the shortest length, while the one that resists more is the power spectrum. I'm going to skip all the other results because I don't have time, but I have results on simulated data which show very similar things. So the intertrial coherence is great when you have a lot of statistics, but it goes down when you have little statistics. And a very last, just a glimpse on the last result that I wanted to show you is that if your aim is not to look at the topography, but just to have a response, of frequency tagging, putting all the sensors together and have a fast and reliable response, we found another measure which is based on canonical correlation analysis. So put all the sensors together, no artifact rejection. It doesn't. You don't need that. All the harmonics together, and then you have a response even at with very short data points. So I'm not going into the details, but in fact, the last result is this one, in which we compare a normalized version of the canonical correlation analysis, which gets significant results even with 4 seconds of stimulation at 1 hertz, so only 4 cycles, while of course the power spectrum decays much faster. So very brief conclusions, just to remind you what I talked about. First of all, I showed you some what I think clear and robust results of using frequency tagging in developmental EEG studies. So in only 1 to 3 minutes of stimulations and with very low frequencies in the delta frequency band, we could answer some important neurocognitive investigations. Then I showed you with the simulations that in fact, while intertrial coherence is more performant with long enough trials, if you go down in statistics, it is more reliable to use the power spectrum or the evoked power spectrum. Then I just mentioned this normalized canonical correlation analysis as a sort of a reliable biomarker measure for applications of any kind. No artifact rejection. This is surprising but true. And lastly, what I was saying before, in this very difficult and challenging sector, it is fundamental to investigate the reliability of the methods that we use. And so I'm convinced that we should do that both by using adult data sets that are longer and more clean, etc., and simulations so that we know more clearly what we are talking about. Thank you.
We have time for a couple of short questions.
About the 3 measures,
yes.
or rather the fact that amplitude measures and phase measures might be different neurophenomena. If you think about sort of the predictive coding framework or something, you would expect phase, and there is evidence to suggest that phase would speed up because of prediction, right? So in a certain sense, if you're just at the low level, sort of stimulating the visual cortex, that's one thing. But if you're looking at higher level things that, say, language, we know that we predict. And so phase would speed up, and that would, first of all, mess up your measures to some extent, but also it's something you might want to capture because it's actually a measure of top-down processing, higher level or whatever, right? So I'm wondering if, in addition to just what you're presenting as their reliability, they actually have different interpretations or potentially different uses. One more of an entrainment kind of thing measure, and the other more of a prediction type of measure.
Yeah, sure. Okay. I think that you raised an important point, because here, what I showed in particular in the methodological investigations is very simple stimuli and with sort of sensory responses. But of course, you're right, amplitude and phase might signal very different processes. And if on one sense, it tags what happens in time in the stimulus. On the other hand, if you also, I think, have to be careful about how much this the temporal profile of the stimulation goes on with the pathway. So when you start from sensory responses, okay, they are phase locked to the stimulation, but if you go to a higher order processing, they might not follow all this temporal profile. And that's why on some cases you might lose the phase and see, for example, induced oscillations that you don't see with phase analysis. So in other studies, we saw that with word reading, some aspects are coded by the amplitude and some by the phase. And I think this can be a matter of bottom-up towards top-down effects. So of course, but this of course is important because not all the results that we find with some checkerboard can be valuable for the rest. I think that the important thing here is to have a sort of model of what you're expecting, put forward a hypothesis and then test them, as you did, in fact.
Studies where true study is applied to cognitive items that are presented in different people. So are there this kind of paradigm that can with a single center?
So there are studies that show simultaneously, for example, faces and houses at 2 different frequencies, and sometimes, and then your perception switches between the 2, and then frequency tagging is able to track this perception. But at the same time, I mean, if you look at the simultaneous, this I do not. I mean, it's difficult also to imagine how you can integrate them at what level. But yes, I mean, one of the seminal studies of, in fact, one of the seminal studies of frequency tagging was one with binocular library. So with the 2 stimuli in the 2 eyes presented at slightly different frequencies, and the result there was that you could track the perception.
Thank you, Marco.
Thank you.
We are now going to try and connect with Stefania. Stefania, ci senti?
Yes, I can hear you. Can you guys hear me?
Yes.
Ok,
Maybe now you can also see me.
Ok, sì, no, ma credo che ci voglia un minimo. Cioè, almeno prima ci va a pensare un po'.
We are a bit late since we had some technical problem.
Tipo anche prima, quando mi hai fatto cost, cioè, ho dovuto aspettare qualche minuto. E se fai conto, su che quanti minuti devi fare?
It's better to use some. I sent
Ma di swamp poi ci vuole.
Just a few moments and ready to go.
vorrei anche che non lo facesse perché come siamo live. Stop recording.
Stoppi te o stoppi? Lo stop.
No.